Welcome to today's lecture on fatty acid biosynthesis. In this session, we will delve into the intricate details of the sequential steps involved in the synthesis of fatty acids in living organisms. Fatty acids are essential biomolecules that play crucial roles in numerous physiological processes, including energy storage, membrane composition, and signaling. Understanding the mechanisms behind fatty acid biosynthesis is fundamental to comprehending the complexities of lipid metabolism and its implications in health and disease. Fatty acid biosynthesis is an enzymatic process that converts simple building blocks into long-chain fatty acids. It occurs primarily in the cytoplasm of animal cells, while in plants and bacteria, it occurs in the plastids and cytoplasm, respectively. The process is tightly regulated to match the cellular demand for fatty acids and is divided into several distinct steps. The first step in fatty acid biosynthesis involves the conversion of glucose or other carbon sources into acetyl-CoA. Acetyl-CoA serves as the substrate for fatty acid synthesis and is generated by various metabolic pathways, including glycolysis, the citric acid cycle, and amino acid catabolism. Furthermore, the conversion of pyruvate to acetyl-CoA is catalyzed by pyruvate dehydrogenase complex, which occurs in the mitochondria. The fatty acid synthase, FAS, complex is a multimodular enzyme involved in the elongation of acetyl-CoA onto a growing fatty acid chain. This complex comprises several enzymatic domains, including acyl carrier protein, ACP, ketoacyl synthase, KS, ketoacyl reductase, KR, and enol reductase, ER. Each domain catalyzes a specific chemical reaction in the iterative process of elongating the fatty acid chain. During fatty acid biosynthesis, the acetyl-CoA units are successively attached to malonyl-CoA by the FAS complex, resulting in the elongation of the growing fatty acid chain. This elongation occurs through sequential condensation, reduction, dehydration, and reduction steps performed by the KS, KR, ER, and ACP domains, respectively. The repeated cycles of these reactions lead to the production of palmitic acid, a crucial product of fatty acid biosynthesis. The production of NADPH is an essential requirement for fatty acid biosynthesis. This reducing equivalent is necessary for the reductive reactions that occur during fatty acid synthesis. The primary source of NADPH in fatty acid biosynthesis is the pentose phosphate pathway, PPP. Glucose 6-phosphate, generated from various carbohydrate precursors, is converted to ribulose 5-phosphate and subsequently enters the oxidative phase of the PPP, producing NADPH. Fatty acid biosynthesis is tightly regulated by multiple mechanisms to maintain cellular homeostasis. Several key enzymes involved in the process are regulated by a combination of allosteric control, covalent modification, and gene expression. Hormones and nutritional status play significant roles in modulating the activity of these regulatory enzymes, ensuring that fatty acid synthesis occurs when required and is limited during times of energy surplus. The acyl carrier protein, ACP, is a central player in fatty acid synthesis as it carries the growing fatty acid chain during elongation within the FAS complex. ACP undergoes a series of reactions involving phosphopentate, covalently linked to a serine residue, and acts as a flexible arm, shuttling the intermediates between all the enzymatic domains of the FAS complex. Desaturation of fatty acids involves the introduction of double bonds into the hydrocarbon chain. This process occurs in the endoplasmic reticulum and is catalyzed by desaturase enzymes. These enzymes introduce double bonds at specific positions, leading to the synthesis of unsaturated fatty acids. Unsaturated fatty acids are crucial for membrane fluidity and serve as precursors for various bioactive lipid species. Fatty acid biosynthesis exhibits stereospecificity, meaning the reaction selects specific stereoisomers with particular bond orientations. This specificity ensures the synthesis of functionally relevant fatty acids and helps maintain the structural integrity of cellular membranes. The enzymes involved in fatty acid biosynthesis display high selectivity towards specific substrates and catalyze stereospecific reactions. Fatty acid biosynthesis is tightly interconnected with other lipid metabolic pathways. The newly synthesized fatty acids serve as precursors for the synthesis of complex lipids, such as phospholipids, triglycerides, and cholesterol esters. Additionally, fatty acid oxidation, occurring primarily in mitochondria, acts as a counterbalance to fatty acid synthesis, maintaining a delicate equilibrium in lipid metabolism. Apart from the canonical fatty acid biosynthesis pathway, specialized fatty acid synthesis occurs in certain tissues and organisms. For instance, polyunsaturated fatty acids, PUFAs, such as omega-3 and omega-6 fatty acids are derived from dietary sources and undergo elongation and desaturation processes. Moreover, specialized pathways exist for the biosynthesis of branched-chain fatty acids and unusual fatty acids like mycolic acid in mycobacteria. Understanding the intricacies of fatty acid biosynthesis has important implications in pharmacology and therapeutics. Various pharmaceutical interventions aim to target key enzymes involved in this pathway to modulate lipid metabolism and treat diseases like obesity, diabetes, and hyperlipidemia. Furthermore, the development of selective inhibitors and activators of specific enzymes opens up therapeutic avenues for various metabolic disorders. In conclusion, the synthesis of fatty acids is a complex and highly regulated process that plays a vital role in cellular function. Through understanding the detailed steps involved in fatty acid biosynthesis, we can appreciate the importance of these biomolecules in health and disease. Moreover, the interplay between fatty acid biosynthesis and other lipid metabolic pathways offers therapeutic opportunities for improving human health.